A toad swallowed a Bombardier Beetle whole in a controlled laboratory experiment. Researchers watching the experiment expected that to be the end of the story. The toad sat completely still for a brief moment after swallowing the beetle. Then its body began to convulse in repeated rhythmic contractions. Approximately 20 minutes after swallowing the beetle, the toad vomited. The beetle walked directly out of the vomited material onto the surface. It was alive and showed no visible signs of injury or distress. It was completely unharmed by the entire experience inside the toad. It crawled a few centimeters away from the ejected material on its own legs and resumed completely normal behavior as if nothing had happened to it. The researchers placed the same beetle back in front of the toad and repeated the experiment. The result was identical to the first trial in every observable way. The beetle was swallowed a second time by the same individual toad. The toad vomited again within a time window similar to the first trial. The beetle walked out of the vomit and moved away again under its own power. Today we are talking about the Bombardier beetle, specifically beetles of the genera Brachinus and Phoropsophis among others, an insect that has evolved one of the most sophisticated chemical defense systems ever documented in any living organism across any taxonomic group, and one that functions just as effectively inside a predator's stomach as it does against threats in open air before any swallowing occurs. Let's start with what the Bombardier beetle actually is as an animal. Before we get into the chemistry and the physics of what it does, Bombardier beetles are not one species. The name covers a large functional group. The name refers to a large group of ground beetles in the family Carabidae, united by the shared possession of the same explosive chemical defense mechanism. There are approximately 500 species distributed worldwide across every continent except Antarctica. They are found in temperate forests, tropical regions, grasslands, desert margins, and agricultural land. Most species are small, between 5 and 30 millimeters in body length. Dark brown or black on top, sometimes with orange or red markings on the head and legs. They are ground-dwelling hunters that prey on small invertebrates. Hunting ants, insect larvae, and other soft-bodied invertebrate prey. They hunt primarily at night and hide under rocks, logs, and leaf litter during daylight hours. Viewed from the outside, the Bombardier beetle looks completely unremarkable. It does not look like a creature carrying what is functionally a small pulse detonation engine in the posterior section of its abdomen, invisible from outside. Now let's talk about the chemistry, because this is where the real engineering lives. Because the chemical system the Bombardier beetle uses is genuinely remarkable from an engineering standpoint. Inside the beetle's abdomen are two chambers separated by a muscular valve. The first chamber, called the reservoir, contains two compounds stored separately, specifically hydroquinones and hydrogen peroxide stored in separated compartments. These two compounds are produced by specialized secretory cells inside the reservoir, and they are kept separate from each other and stable at normal conditions. The second chamber, called the reaction chamber, is smaller and more muscular. Its walls are lined with enzymes, specifically peroxidase and catalase. These enzymes are the critical component of the entire system. When the beetle is threatened, it relaxes the muscular valve connecting the two chambers, specifically hydroquinones and hydrogen peroxide stored in separated compartments. When the valve opens, they flow together into the reaction chamber. When they contact the enzymes in the reaction chamber, the reaction begins immediately. The hydrogen peroxide is catalyzed into water and oxygen gas. The oxygen then rapidly oxidizes the hydroquinones into benzoquinones. The oxidation reaction is highly exothermic. It releases a significant amount of heat very rapidly. The heat combined with the oxygen gas generates enough pressure that the mixture is expelled from the tip of the beetle's abdomen as a hot gas. The temperature of the expelled mixture at the point of discharge is approximately 100 degrees Celsius, exactly the boiling point of water at standard atmospheric pressure, 100 degrees Celsius. This is not a rounded approximation. Measured temperatures at discharge reach exactly this. The chemical reaction generates sufficient heat to reach the boiling point of water. At the moment, the mixture contacts the enzyme lining of the reaction chamber, and the mixture that exits the beetle is genuinely at that temperature. At the moment, it makes contact with whatever is touching the rear of the beetle. Now let's talk about the discharge mechanism in greater mechanical detail. Because the temperature is only the first remarkable aspect of how this system works. The discharge from the Bombardier Beetle is not a single continuous chemical spray. It is a rapid series of discrete pulses fired in extremely fast succession. High-speed photography and acoustic analysis of Bombardier Beetle discharges have measured the pulse rate at between 368 and 735 pulses per second, depending on the species studied. This means the beetle is firing its chemical weapon hundreds of times per second. The pulsing behavior is not a deliberate decision the beetle makes during discharge. It emerges automatically and inevitably from the physical mechanics of the reaction chamber. When the beetle fires, the pressure in the reaction chamber drops momentarily after each pulse. This pressure drop causes the muscular valve to close briefly. The pressure drop closes the valve and cuts off flow from the reservoir briefly. The reaction chamber then repressurizes from the ongoing reaction. When pressure rebuilds, the valve opens and the next pulse is expelled in sequence. This is a completely self-regulating mechanical system requiring no active neural control. The valve opens and closes automatically in response to pressure changes, without any active muscular control during the firing sequence beyond the initial opening. It is mechanically similar in principle to a pulse jet engine, where fuel and oxidizer mix react and are expelled in rapid pulses driven by pressure cycles. Pulse jet engines were used in several experimental aircraft designs in the 20th century, and researchers studying the Bombardier Beetle's mechanism have noted the structural similarity between the Beetle's discharge system and the operating principle of a pulse detonation engine. The Beetle evolved this mechanism approximately 250 million years ago, hundreds of millions of years before any human aircraft engine existed. And the engineering solution it arrived at is structurally similar to what human engineers independently developed by human engineers for a completely different technological application. When the Beetle fires, the sound is audible to the human ear without amplification, a rapid clicking or popping sound that can be heard from a short distance. This sound is produced by the rapid pressure pulses as each discharge occurs. It serves as an auditory warning signal to predators that have learned that this particular sound is associated with an unpleasant chemical experience. Now let's talk about the aiming system, because the Beetle does not simply spray blindly. Because the Bombardier Beetle does not simply spray in one fixed direction. It aims the discharge precisely toward whatever is threatening it. The tip of the abdomen where the discharge occurs can be rotated, both downward and laterally, to cover a significant range of directions. The Beetle can direct the spray toward its own front, toward its sides, or direct it almost directly backward at a threat approaching from the rear. Researchers testing the Beetle's targeting have found that it consistently aims toward the precise point on its own body where physical contact with a threat occurred. If a predator grabs the Beetle's left leg, the discharge is directed to the left. If it is grabbed from the front, the abdomen rotates to direct the spray toward the front. This targeting behavior is reflexive rather than deliberately aimed in any cognitive sense. The Beetle does not appear to make a deliberate aiming decision. The response is triggered by the location of the physical contact, and the abdomen rotation is automatically directed toward the contact point. The speed and accuracy of this targeting response means that a predator grabbing any part of the Bombardier Beetle is very likely to receive the discharge directly on precisely the body part of the predator that made contact with the Beetle. Now let's talk about what the discharge actually does to a predator. Because 100 degrees Celsius at the point of release sounds lethal, but the amount of material involved is small and the Beetle is also small. The primary effect of the discharge on a predator like a bird lizard or frog is an intense burning and irritating sensation from the benzoquinones, combined with a startle response from the audible click and the sudden thermal contact. The benzoquinones are specifically irritating to mucous membranes. The soft tissue inside the mouth, throat, and eyes is particularly sensitive to these compounds. For most small predators, this combination of chemical irritation, thermal shock, and auditory surprise is sufficient in most cases to cause immediate release of the beetle by the predator. The predator drops the beetle and the beetle runs or flies away. Researchers have tested the effectiveness of the discharge against multiple predator types. Spiders, birds, lizards, frogs, and various insect predators have all been tested. In the majority of cases, the discharge is effective at causing release and escape. It is not universally effective. Some large or experienced predators can sometimes successfully consume and retain a Bombardier beetle without ejecting it, but the failure rate is significantly lower than for undefended beetles of comparable size, without chemical defenses. Now let's get to the detail that the toad experiment reveals, which is the fact that the defense works inside a predator's digestive system. The 2018 study conducted by researchers at Kobe University in Japan tested whether Bombardier beetles could survive being swallowed by toads. They offered Bombardier beetles to Japanese common toads Bufo japonicus and observed what happened in each trial with careful documentation. In 43% of the individual trials, the toads successfully swallowed the beetle. In every case where the beetle was swallowed, the toad vomited within 44 minutes. In the majority of trials, within a window of 12 to 35 minutes after swallowing. The beetles that emerged from the vomit it was alive and showed no visible signs of injury or distress and appeared unharmed. Control beetles of similar size without the chemical defense, that were fed to the same toads as a control condition for direct comparison, were not vomited out and were retained and digested normally. They were simply digested without any vomiting response from the toad. The discharge events inside the toad's stomach created sufficient thermal and chemical irritation of the stomach lining, sufficient to trigger the toad's vomiting reflex and expel the beetle. The beetle was expelled before the digestive process could kill it. The researchers found that the beetles discharged multiple times inside the stomach. The researchers detected discharge events using acoustic sensors monitoring for clicks, specifically the audible clicking pattern produced by each individual pulse discharge, detectable through the toad's body wall from outside without invasive measurement. The beetle fired its chemical system multiple times over the period it was inside the stomach, and the cumulative effect of these discharges was sufficient to cause ejection. The researchers noted that this is a genuinely unusual defensive scenario. Most chemical defense systems in insects are pre-contact defensive systems, warning coloration or pre-warning secretions that deter attack before consumption occurs. The Bombardier beetle's defense is specifically post-ingestion in its most extreme application. It functions after the worst-case scenario for prey has already occurred, and it successfully reverses that worst-case scenario in a significant percentage of cases. Now let's address the evolutionary history of the Bombardier beetle's defense, because the mechanism is complex enough that it raises the obvious question of how something this precisely calibrated could evolve incrementally. The Bombardier beetle's defense has been cited by proponents of intelligent design as an example of irreducible complexity, the argument that some systems are too complex to evolve step by step, because removing any component makes the whole system non-functional. Biologists studying carabid beetle evolution have addressed this argument directly by examining the chemical defenses of related beetle species that do not have the full Bombardier system. Several related carabid beetles produce quinone secretions without the explosive discharge mechanism. These beetles simply release quinone compounds from their abdominal glands when threatened. The secretions are noxious and provide some deterrent against predators. But these related species produce secretions that are neither heated nor pulsed. Other related species produce hydrogen peroxide secretions without the full reaction chamber system. And still other related species show intermediate states of the full reaction chamber system, with some enzyme activity but less developed reaction chamber structure. Taken together, these related species provide a plausible evolutionary pathway, from simple ancestral quinone secretion through measurable intermediate stages to the fully developed pulsed explosive discharge seen in Brachyna species. At each stage, the intermediate version of the system provides some degree of predator deterrence even without the full mechanism. This makes the Bombardier beetle's system consistent with incremental evolution, where each intermediate stage provides measurable predator deterrence even without the full system, rather than requiring the complete system to appear fully formed in one step. Now let's talk about what happens to the beetle between the swallowing and the vomiting, because the toad experiment did not allow direct observation of what happens inside the stomach, and represents a genuinely interesting gap in what we currently understand. The researchers could detect discharge events from outside the toad, using acoustic sensors that picked up clicks through the body wall using acoustic monitoring. But they could not directly observe what was happening to the beetle inside the stomach. What we know is that the beetle survived and appeared unharmed after ejection. What we do not know is whether the beetle was conscious during the stomach period, whether it was simply responding to the stomach acid and muscle contractions with discharge, or whether some degree of protective secretion from its own body contributed to its survival. The beetle's cuticle, the outer shell, is presumably providing some protection from stomach acid, and the beetle's ability to continue discharging inside the stomach suggests its biochemical systems remained functional under those conditions. But the specific physiological experience of a beetle inside a toad's stomach for 12 to 35 minutes, and the precise mechanism by which the beetle survives intact during that time, are not fully characterized or understood in the currently published research literature. This gap is itself interesting. We have a documented outcome that seems almost impossible, and a partial but incomplete mechanistic explanation of how it happens, which is exactly the situation that generates the most productive scientific research. Now let's also discuss something about the chemistry that connects to human applications. Benzoquinones and related compounds produced by Bombardier beetles and related carabid beetles have attracted research interest for potential antimicrobial pharmaceutical applications. Quinone compounds generally have significant antimicrobial activity, and the specific compounds produced by various beetle species have attracted interest from researchers looking for new antimicrobial agents. The mechanism by which quinones damage bacterial cell membranes is different from the mechanisms used by most current antibiotics, which makes them interesting as potential treatments for antibiotic-resistant bacteria. This research avenue is currently at a relatively early stage of development, but it represents another case where an evolved chemical system in an insect turns out to have properties of direct practical interest to human medicine. The Bombardier beetle evolved its quinone production system as a predator deterrent, and those same compounds happen to interfere with bacterial cell function in ways that makes them worth investigating as potential treatments for resistant bacterial infections. Write in the comments if you had seen video of a Bombardier beetle firing before today, because the high-speed footage of the discharge is one of those things that is genuinely difficult to believe even while you are watching it. A beetle being grasped by laboratory forceps in a controlled experimental setting, producing a rapid, audible series of popping clicks from the tip of its abdomen, accompanied by a visible puff of gas expelled at the point of each pulse. While remaining completely, it was alive and showed no visible signs of injury or distress, and apparently undamaged by a reaction happening inside its own body, reaching 100 degrees Celsius per pulse. The beetle has been running this exact chemistry for approximately 250 million years. It got the engineering right long before anyone was there to watch. There is a specific study from the 1960s that established much of what we know about Bombardier beetle behavior. Thomas Eisner at Cornell University began systematically studying Bombardier beetle chemistry and behavior, and published foundational work that forms the basis of most subsequent research on this species group. Eisner's approach combined behavioral observation with chemical analysis, documenting both what the beetles did when threatened and what compounds they were producing. The hydrogen peroxide and hydroquinone chemistry was characterized in Eisner's laboratory, and the explosive nature of the discharge was demonstrated using both direct observation and high-speed photography. The pulse jet mechanism was not fully characterized until later work using acoustic analysis, but Eisner's foundational studies established the framework within which that work became possible. Eisner continued studying chemical defenses in insects for decades, and the Bombardier beetle remained one of his central subjects throughout that research program. His work contributed to the broader field of chemical ecology, the study of how chemical compounds mediate interactions between organisms in ecosystems. The Bombardier beetle is now one of the most studied examples in this field, precisely because its chemistry is so dramatic and so precisely characterized. The clear connection between specific chemical compounds and specific behavioral outcomes makes it a useful model for understanding how chemical defense systems function generally. Now let's talk about something specific that the toad vomiting experiment revealed. Beyond the simply documented fact that the beetle survives intact, the timing of vomiting in the toad experiment was not random. In the experimental trials where individual beetles survived being swallowed, the vomiting occurred earlier when the beetle was more recently swallowed. and in some cases occurred faster than in others across multiple trials. The researchers interpreted this variability as reflecting variation in discharge activity. Beetles that discharged more quickly or more intensively inside the stomach produced the toad's vomiting response earlier than less active beetles. This suggests that the beetle's behavior inside the stomach, specifically how actively it discharges, is a variable response that differs between individuals and conditions, and that this variability may reflect individual beetle condition. Beetles in better condition with more chemical reserves may discharge more intensively, producing faster vomiting and therefore spending less time in the stomach, which would reduce the risk of injury from stomach acid and muscle contractions. This interpretation is speculative and based on a relatively small number of trials, but it raises the interesting question of whether beetles that have recently discharged multiple times, and therefore may have significantly depleted chemical reserves available for discharge, would survive swallowing as reliably as beetles with full reserves. The chemical system requires time to recharge after discharge. The two compounds in the reservoir are produced by secretory cells, and production is a metabolic process that takes time and resources. A beetle that has recently fired multiple times in response to threats may have partially depleted reserves until production catches up, and may therefore have less discharge capacity available if immediately swallowed after multiple prior discharges. Whether this actually affects survival outcomes in the stomach has not been directly tested. It represents a real and testable question about the operational limits of the discharge system. Now let's talk about the broader family of beetles that use quinone chemistry. Because the Bombardier beetle sits within a much larger group of chemically defended insects, the ground beetle family Carabidae is one of the most species-rich beetle families on Earth. The family contains approximately 40,000 described species across all regions of the world, and chemical defense is widespread across many of its subfamilies. Most carabid beetles produce quinone secretions from abdominal glands, including species that are not classified as Bombardier beetles. The secretion in non-Bombardier carabids is typically a simple release of compound, without the explosive heated discharge that gives Bombardier beetles their name. But the basic chemistry, quinone compounds with deterrent effects on predators, is shared to varying degrees across a very wide range of carabid beetle species. This widespread distribution of quinone defense across carabid beetles supports the incremental evolutionary model discussed earlier in this account, where the Bombardier beetle's mechanism represents a refined elaboration of a chemical defense system that was already present in ancestral carabid beetles, rather than a completely novel system arising without precursor. The 500 or so Bombardier beetle species represent the extreme end of a continuum, at the other end of which are species with simple passive quinone secretion, and in the middle are species with various intermediate degrees of chemical defense development. This continuum visible in living relatives is strong evidence that the Bombardier mechanism evolved through gradual incremental steps with intermediate stages, each providing some defensive value even without the complete explosive discharge system. Now let's talk about what Bombardier beetles do in their daily life when they are not actively defending themselves against predators including toads, because the explosive chemical defense is their most famous and studied feature. But that defense represents only one part of a complete ecological life history. Bombardier beetles are active nocturnal hunters during most of their lives. They move through leaf litter and surface soil searching for invertebrate prey, using chemosensory organs in their antennae to detect prey at short range. Combined with visual detection of movement at slightly longer distances, they are most active at night when their primary predators, birds, are least active. During daylight hours, they shelter under rocks, fallen bark, and other ground-level objects, emerging after dark when bird predation risk is substantially lower. Their prey typically consists of smaller, soft-bodied invertebrates found in soil and litter, insect larvae, earthworm segments, small beetles, springtails, and worker ants. The ants deserve specific mention because many Bombardier beetle species are found in habitats with high ant density, and ants are one of the most likely animals to encounter a ground beetle in their territory. The Bombardier beetle's chemical discharge is highly effective against ant attacks. Ants that contact the discharge are repelled and in some cases incapacitated temporarily by the combination of chemical irritation and thermal contact. Individual ants can sometimes mob a bombardier beetle in sufficient numbers to overwhelm the discharge rate if the beetle cannot turn and target effectively. But in most individual ant versus beetle encounters, the beetle's defense is decisive. The bombardier beetle also has a chemical vocabulary beyond the defensive discharge. Like many ground beetles, it produces sex pheromones for mate location and trail substances that may help it navigate between shelter sites and hunting grounds. These chemical communication systems are less studied than the defensive discharge, but they represent an important part of the animal's behavioral repertoire. Now let's address something about the physics of the discharge that was only fully characterized in mechanistic detail relatively recently. When researchers began using synchrotron X-ray imaging to observe the discharge in real time, synchrotron facilities generate extremely intense X-ray beams suitable for high-speed imaging that can be used to generate high-speed X-ray movies of small organisms. Researchers used this technology to image a live bombardier beetle during a defensive discharge and observed the actual mechanics of the reaction chamber and valve system operating in real time at full speed during an actual beetle defensive discharge event. What they found confirmed and extended the acoustic and chemical analysis of earlier decades. The reaction chamber wall was observed expanding and contracting with each pulse, as internal pressure built and the valves cycled open and closed. The sequence of valve opening, chemical injection, reaction pressure buildup, and expulsion was directly visualized at a level of detail that previous techniques could not achieve. This work was published in 2015 and represented the first direct visualization of the internal mechanism operating during an actual discharge event. It confirmed the pulse jet model that researchers had inferred from acoustic and chemical evidence, and demonstrated that the beetle's mechanism functions with a precision that matches the theoretically predicted mechanical model almost exactly. This kind of direct visualization of a biological mechanism at this speed and resolution represents the cutting edge of what is currently possible in entomological imaging. And the bombardier beetle was a sufficiently interesting subject to justify the use of a synchrotron facility for a beetle that most people would never notice, walking along a forest path that passes over the leaf litter where it lives. Write in the comments if you had previously heard of the bombardier beetle before today, because for me this is one of the clearest examples in biology of something in biology that sounds entirely impossible until examined carefully. Until you look at the evidence and find that the evidence is extraordinarily thorough. A beetle that heats a chemical reaction to the boiling point of water inside its own body, fires the result hundreds of times per second with audible sound, aims that discharge precisely toward whatever body part of the predator grabbed it, and survives being swallowed by a toad by firing the same system inside the toad's stomach. The toad vomits because the beetle fired its system inside the stomach repeatedly. The beetle walks away from the ejected material on its own legs completely unharmed. And this specific defense system has been operating for approximately 250 million years. There is one more detail about how the bombardier beetle's defense works over time that connects back to the broader question of what makes a defense system evolutionarily stable. The bombardier beetle's chemical defense is metabolically expensive to maintain. Producing hydroquinones and hydrogen peroxide in the reservoir requires metabolic energy. The enzymes lining the reaction chamber require production and maintenance. The muscular valve and the precision-engineered reaction chamber walls are structures that take biological resources to build and maintain. All of this investment must be worth it in terms of survival advantage, or the energy would be better spent elsewhere, on reproduction or growth. The high frequency with which the defense is effective against predators, and the dramatically lower predation rate on bombardier beetles compared to undefended beetles of similar size, confirms that the cost is outweighed by the survival benefit. But the defense also has limits, and those limits reveal something interesting. The amount of chemical reserve available for discharge is finite. A beetle that has discharged its system many times in rapid succession eventually exhausts its reserve and cannot fire again until the reservoir refills. Refilling the reservoir through secretory cell production takes several hours. During that refilling period, the beetle is effectively without chemical defense. Researchers have shown that a beetle whose reserves are depleted does not produce the discharge and can be consumed by predators that would normally be deterred. This vulnerability window is essentially invisible from the outside. A depleted beetle is physically indistinguishable from a fully charged beetle from the outside, which means the defense works partly on the reputation of the species as a whole. A predator that has learned to avoid bombardier beetles from prior experience will avoid any individual that visually matches its memory of a bombardier beetle,